Hello, and welcome to this talk. My name is Tony. I am a third-year PhD student at the Department of Chemical Engineering and Biotechnology. And today I'm going to tell you about my research in bioenergy and how I use AI, artificial intelligence, and machine learning specifically to speed up some of my experiments. So let's go on to the slides. I'd like to begin talking about the motivation for my research. And for that, I'd like to show you one of my favorite photos. This is the largest solar floating PV farm. It's based in Huainan in China, and it sits on an artificial lake, which is above an abandoned coal mine. The coal mine represents a vice that we need to move away from as human society, which is producing electricity from fossil fuels. And one way we've been doing this since the 1950s is by using solar PV panels, which you can see floating above the lake. And the solar PV panels have been critical in terms of helping us decarbonize our electricity system. They use sunlight energy and the photon energy from the sunlight to generate an electric current. However, we see also there's this islands of green all across the lake, and these are plants or photosynthetic organisms that have been doing this for over 3.5 billion years. They survive out of photosynthesis, and they use sunlight energy to generate energy for their own use. And so we can potentially harness the process of photosynthesis to generate electricity for our own needs. And so for the next 20 minutes, let's explore how we can actually harness photosynthesis for electricity generation. What are some of the challenges which are slowing down our progress to date? And how we can use artificial intelligence to help speed things up. First, a quick review about photosynthesis and how it works. Quite a wide range of organisms perform photosynthesis, all the way from cyanobacteria at the micrometer scale, all the way to trees, which can be tens and up to even 100 meters tall. What they do is they use the photon energy from the sun to strip electrons from water in what is called the light reactions. And they use the energized electrons and combine these with carbon dioxide that they absorb from the atmosphere to generate sugars, which they store as chemical energy for later use. At night, when it's dark, the reverse happens. They can use the sugars that they've produced during the day and convert this back to energy for their own use, and at the same time generating electrons that we can harness for our own use in electrochemical devices. In my research, I use cyanobacteria because they're simple single-celled organisms with a single strand of DNA, which makes it easy or a lot easier to understand what's happening within them compared to maybe algae or plant life. We see in some cyanobacteria that they're able to donate the electrons that they generate during photosynthesis, either immediately or later on during respiration, and donate these electrons to their surroundings. This might be a way for them to obtain important nutrients from the environment, for example, iron, which is quite critical for the photosynthetic machinery. And it might also be a way for them to communicate with each other through a chemical language, and this has a fancy name called quorum sensing. But irrespective of the motivation for donating electrons to the environment, we can capture some of these electrons in electrochemical devices known as biophotovoltaic devices and generate electricity for our own use. The schematic on the left shows what a photovoltaic device looks like. It's a simple device split into two halves. The first half is called the anode, and this is where we host the photosynthetic organisms. And this is where, once they're hit by sunlight, they can donate electrons to the anode. And the electron can then travel across an external circuit to the second half of the device, which is called the cathode, where it recombines with oxygen from the air to produce water as your only byproduct. So it's a relatively clean and simple process with water as your only byproduct. You might be wondering how these devices look like in reality, and for that there's very many different devices that people use. My own design is shown on the right here. It's a simple bottle, and where we have a red box highlighted here is where a lot of the electrochemical reactions are occurring. If you zoom in to that red box, We see here the anode side of the biophotovoltaic device. And this black circle here is carbon paper, which is a typical material used for the anode because it allows the biofilm to grow quite happily. And this slightly darkened part of the carbon paper is where some cyanobacteria biofilms have grown. And if we actually zoom in further into that section, we can see here a scanning electron micrograph of some of the cells growing on top of the carbon paper. At the moment, the performance of these devices are quite poor. They're around 1%. And relative to traditional solar panels that you might see at your neighbor's house or driving along the highway, this is significantly lower. If you look at the actual output of power, it's around 3 orders of magnitude lower in the biophotovoltaic device compared to a traditional solar panel. So we do have a long way to go. One of the main challenges is understanding how these electrons flow from within the organism onto the anode. We don't currently have a good enough understanding of how the electrons are traveling from within the cell to the anode. And we have a few ideas around this. One is that it's an indirect process, in which case the bacteria release some either chemicals or proteins, which have electrons, which then travel all the way to the anode, and then are deposited onto the anode. And then the shuttles can then travel back to the cell and collect more electrons. A second way we think this might happen is through direct transfer using nanowires. So nanowires can be thought of as extensions of the cell membrane, which then go unattached directly onto the cell, onto the anode. And because they're conductive, the electrons can flow directly from the bacteria to the anode. The last potential approach might be that you have a direct transfer from the cell membrane onto the anode, but it could also be a mix of all three. And one way we study the potential different ways that the bacteria are transferring electrons to the anode is looking at how they behave when we stress them in extreme environments. We do this by, for example, putting the bacteria in extreme levels of carbon dioxide, or putting them in extreme, or deriving them of important nutrients such as iron, as I mentioned, which is important for the photosynthetic machinery. However, this is quite a slow process. A typical experiment which tries to look at how a particular set of environmental factors affect the performance of the device takes up to 2 weeks long. It starts off with growing the bacteria in the particular environment you want to test, followed by assembling the device, preparing the device by growing the biofilm on the anode, conducting your experiments, and cleaning up. And 2 weeks for conducting one set of experiments is quite a long time. And so we can look at how we can use machine learning to help speed this up. So let's look at how artificial intelligence can help us do this. First, artificial intelligence is quite a large and broad term, and a subset of artificial intelligence technologies are artificial neural networks. These are loosely inspired by the human nerve cell in terms of the structure, but I'd like to make it clear that they do not work in the same way. But what artificial neural networks are quite good at is predicting a particular output from a set of input factors that you give it. So in my case, I'm kind of interested in how, if I can predict the current output from the BPV from certain environmental factors such as the amount of CO2 in the anode chamber or the amount of iron in the media where the bacteria are growing. So how can we do this in practice? In this example, I'd like to find out what is the best combination of CO2 in my anode chamber and the amount of iron concentration that I should have in the media in which my bacteria are growing. For this, I can choose extreme regions or extreme points where I know the bacteria will struggle in terms of their survival, but they will still survive. For example, I know that the bacteria can survive in up to 20% CO2, and I know they can survive in an environment where there's absolutely no iron in the environment. So I can conduct a few experiments, in this case 4 experiments at the very extremes of this region, and maybe somewhere in the middle of the region. and collect actual performance data from my BPV device. With this few sets of experiments, I have sufficient data to train an artificial neural network to be able to predict the amount of current output based on the amount of CO2 and the amount of iron in my environment. After I've fully trained the BPV device, I can look at the performance and see how well the current output of the BPV is being predicted by the neural network. So on the left is actually quite recent experimental data from a BPV device, and it shows the performance of the neural network in terms of predicting the output of the BPV, both in the light, shown here in the white background, and in the dark, shown here in the gray background. And as you can see, the neural networks shown by the red dotted lines predicts the current output quite well, to the extent that it actually overlaps quite well with the actual experimental data. So now that I'm happy that the neural network is actually predicting the performance of the BPV device quite well, I can then choose any number of different combinations of iron concentration and CO2 concentration and conduct simulations to give me an idea of how well the BPV will perform when I go into the lab and spend 2 weeks conducting an experiment. And so each simulation takes less than a minute, and we can actually obtain quite a lot of data points to see how the performance changes with CO2 level and with iron concentration. In this example, we see that if I set the CO2 concentration to around 15% and I reduce the iron concentration to 0, I get the maximum performance in my device. And so when I go into the lab, I then have a very targeted, specific region that I want to test, instead of going in blind and spending 2 weeks to get each of these single blue points, which would really take, if I was to do this in the lab, over 200 weeks. Another way neural networks have been used in research around photovoltaic devices is to predict the actual power output from live weather conditions. So we have a group in the University of Stanford who use a live video feed of the sky to look at how much sunlight is reaching their solar panels and have an instantaneous prediction of how much power is being produced. And this is particularly useful when you look at scaling up these devices, where the instantaneous power output has a huge impact on the performance of your electricity grid. And so in the future, we might be able to do similar things with BPV device once we have them performing at scale and commercialized. So just to conclude, a few final words of a few key points that I've hoped you've learned today. One is that we can harvest photosynthetic electrons to produce electricity. And the advantage over traditional solar PV cells is that these electrons can be harvested both during the day when the sunlight is shining, and also at night when there is no sunlight, because we can use the same electrons during the respiration process to generate electricity in the dark. Secondly is that we don't have a full understanding of the electron path, which is limiting how well these devices are performing at the moment, but we're working on it. And we can use artificial intelligence to nudge us in the right direction. However, the artificial intelligence does not necessarily replace traditional models. It's a way at the moment that we can speed up experiments. In the future, we may be able to use artificial intelligence in more interesting ways, for example, controlling these devices or predicting power output instantaneously from weather conditions. And finally, I'd like to say that the energy transition requires multidisciplinary solutions. My research includes not just electrochemistry, which is my expertise, but also looking at biology and also looking at computer science. And this is the nature of what the best solutions to the energy challenge that we face in the 21st century will be like. They will require input from everyone, and so everyone has a role. A place like the Department of Chemical Engineering is particularly good for this because it's an inherently very multidisciplinary department where you'll find researchers looking at everything from neuroscience down to traditional thermodynamics. And with that, I'd just like to thank you and thank my funders for my scholarship project, the Cambridge Africa program. And I hope you've enjoyed the last 20 minutes we've spent together. Thank you.